everyone, and welcome to Red Hall, the library chat. I am Francesca Bartolini. I am an associate professor in the Department of Pathology and Cell Biology at Columbia University and a visiting scholar at Red Hall for a couple of weeks. Today I am delighted to introduce you to my good friend and colleague Germano Cecere. I will let Germano introduce himself first and then we'll start with the conversation.
Well, first of all, thank you, Francesca, to invite me here. It's a great pleasure to meet you here in Paris. We have known since a long time in New York. So I'm actually based in Paris. I started my lab in 2016 at Institut Pasteur, where I'm directing a lab of mechanism of epigenetic evidence, where we are studying this new type of heritable information that we can explain basically some other type of disease heritability that where we don't find any clue of what DNA mutation we can find and something like this.
Can you define in very few words the difference between epigenetics and conventional genetic?
Wow, that's a huge debate. Well, yeah, essentially, everything we know about disease heritability is maybe coming from the fact that we think that we inherit some DNA mutation from our parents, and this will lead to some malfunction in the body. But actually, now we know that there are other type of molecules that can also be inherited through what we call gametes, which is the sperm and the oocyte when we are conceiving the newborn. And so these new molecules that we are investigating are on top of the gene. That's why it's called epigenetics from the Greek. And so they might regulate the DNA information containing in our genome without having any changes in the DNA sequences. So there are no mutation associated with genes, but those molecules and the changes in those molecules that can be inherited might lead to some predisposition of diseases or even inherited some features to the next generation.
And I gather that this is very conserved in evolution, correct?
This is extremely conserved and we know now the players. We know that there are certain chemical modifications that are deposited around the DNA. In fact, I didn't tell you that our DNA is not free in our cell. In fact, it has been estimated that it's 2 meters long, this genome that we have.
In humans.
In humans, in every cell. So, these 2 meter DNA fibers should somehow be compacted in our cell to be
in the nucleus.
In the nucleus. And so it's very highly organized around some proteins that we call histones. And so it's wrapped around these histones. Those are like beads. Mm-hmm. And so what we discover, in the past 20 years or more, is that those beads where the DNA is wrapped around, they can be chemically modified by many different modifications. And those are what we call epigenetic modification, which in fact, the combination of different type of modification might lead to some activation or repression of the information contained in our genome.
So it's very combinatorial.
It's a combinatorial messages, and in fact, it has been proposed that there is a code that underlie this all those epigenetic modification, which I think is something related to what you study as well, because what you do.
Yeah, it's true. So I'm not an epigeneticist. I was trained as a biochemist and as a biologist during my postdoctoral studies. But I was always very interested in understanding the biology and the function of a specific cellular player, which we call macrotubules. And I'll tell you very briefly what they are. They look like hollow rods they're filamentous, they can assemble and disassemble, and they provide the major component for what we call the skeleton of the cell. Yes, also cells have a skeleton, which is called the cytoskeleton. And macrotubules do a lot of things. As you know, microtubules are very important for cell division because they actually provide the scaffold for the segregation of the genetic material into the two daughter cells. But they also have a very important structural role for the determination of cell shape, for breaking the symmetry, which is a big concept in cell biology. When a cell, for example, has to change shape from being round and globular to polarized, right, and elongated, and this happens in several cell types. But it has also a very important function in providing sort of a railroad tracks for intracellular transport. And what I mean by that is that they, because of their elongated geometry and shape, they literally act as highway for the bidirectional transport of a lot of molecules and a lot of other intracellular protein and organelles. Organelles are just, little intracellular containers, like factories for whoever is not a biologist with specialized functions. So we discovered maybe a couple of decades ago that these tracks can be modified. They can be modified chemically, just like your histones, right? And the combinatorial nature of these modifications, which are several, can be read by the cars and the tracks that travel onto these highways as either stop signals or go ahead signals or turn right or turn left.
So there might be a specific code.
Correct. In fact, so there is a professor here at the Curie-Orsay, Dr. Karsten Yankee, very dear friend and colleague, who is the person who actually chose the name he invented the name for the tubulin code, and it's called the tubulin code because these elongated filaments, these filaments that we call microtubules, are made up of smaller pieces, and these smaller pieces are called tubulins, and the tubulins are the bricks of these elongated pieces of walls that can be modified, and that's why we refer to it as the tubulin code. The tubulin code is at this point just an hypothesis, the rules of which need yet to be elucidated, but we're discovering that it's very important for the regulation of the many different functions. We wouldn't be us if there wasn't this superimposed combinatorial sort of epigenetic code in tubulin. When we say epigenetic, I say by analogy with him, but it's not really an epigenetic code. It's just like a different kind of code that can be read by different players. And in neurons, this is particularly important because in neurons, as you know, they don't divide. So it's not that they use the tubulin because they need to segregate into different cells, but they are very long. They can be like meters long. And so the railways become essential for having the neurons being able to send a message or any other cellular component from one place to another. And we're also discovering, this is something that I'm doing really, my current work is pretty much based on this. We're trying to understand the rules of the code in neurons. And this seems to be very important in health as well as in disease.
Okay. So what do you think might be this coding potential of all this modification that you have on the tubulin?
Oh my God!
On the heritability.
That's a great question. We have no idea about that. Because these filaments are polymer that undergo constant bouts of assembly and disassembly, it's hard to believe that they can actually persist. Exactly. It's but what you can imagine is that, for example, maternal germinal cells could carry different tubulin modifying enzyme that and maybe that could be something that might skew the one modification into a certain direction during the first division of embryogenesis.
Okay.
And but people really have no idea yet.
So there is a
we could do it together. Let's do it.
So we there is basically a commonality
Yeah.
in our work in terms of what other molecules can be modified in the cell to provide some heritability in my case. I see.
Well, I think in my case, the tubulin code is more for the coordination of complex cellular function. Also because you can imagine that these filaments can carry all these modifications, right? They are decorated by all these different patterns. And so it's almost visually easier to see that there is a code there, like a pattern can be read and can be erased, can be modified. So I think that's the true analogy. I mean, the analogy that we know.
So do they play any role in this genetic material that we are talking about?
Yeah, I mean,
DNA,
some of these modifications are also present on the mitotic spindle. The mitotic spindle is the scaffold that allows, like I said before, the segregation of the chromosomes during cell division. There are some evidence that the tubulin code is also regulating some of these functions. And but obviously I'm not surprised because microtubules are very conserved structures, and they've been doing this for a long time. So
good.
Yet we need to discover a lot.
So what about the context that you study those processes?
In the context or the organism? The context.
Organism context.
Well, I am interested in. So I really think we don't know enough about the rules of the code. So I'm interested in understanding what these rules are. As I said, I was trained as a biochemist, but at the moment I'm really trying to understand how the code regulates physiological functions, because this is something that traditional has never been the aim and the focus of biochemists. They're interested in tubulin biology. And this is a very, very open question because, I'm actually asking, okay, so the little modification that is on those filaments right there in the neuron, how is that translating into allowing formation of memories, for example? How is that translating into the regulation of sensory function? So when you're asking these questions, obviously you have to be ready to bring this your experimental model to a different level. As far as conservation, and then we'll get to your experimental model, but as far as conservation, we in my field, we realize that the highest complexity is in mammals, so we try to stick to mammals, and so ask the question first in health, as what is the tubulin code regulating in normal function? Like, is the tubulin code important for me, for example, to feel the cold or the heat? Is it important for me to touch? And so in sensory function. And then, of course, we also know, and we're very interested in understanding whether this function of the tubulin code is something that also underlies a neurodegenerative disease. And there is an emerging area of investigation that aims at addressing this question.
I see.
What about, in other words, I'm working with small mammals, rats and mice, and animal models of both neurodegenerative and neuropathic disease. But I know that you work with a far more interesting animal, right? So tell us a little bit about it.
Well, I wish to have it here, but I don't. There is a very important point. In your case, you're studying this code of tubulin. In our case, we really need to study a code that is inherited.
Right.
And so to avoid that, we always run into genetic changes. We are all different. We are all coming from different parents. So we need to choose an organism where the DNA is absolutely not changing across generation of individuals. And so we were the fortune to meet this little organism that was chosen a long time ago for other type of studying biology.
By Sidney Brenner, right?
Yes, and it's called Caenorhabditis elegans, C. elegans, because it's very elegant in the way he moves.
Can you show us?
I show you. It moves like this. That's the only thing I can
Is that where the elegance comes from?
It's
okay.
so small that in fact you cannot see by eyes. So you need a microscope.
Okay.
It's transparent, but the important feature that we like is that it's one of the few animals that is self-fertile hermaphrodite. So meaning that the same animal has both sex and it can reproduce without the help of any others.
Very handy.
Very handy. And is very fertile, is making like 300 babies in only 3 days. So we have, extremely fast generation times, which is useful if you want to study process of multiple generational. That we can do in few weeks. We have 3-4 generations.
So give us a small example.
And the other important thing is that is they are all identical.
Right.
So there are no genetic changes in the population. This is very important. So we can really study processes that are heritable in this organism, and we are sure that nothing changes in their DNA code. And maybe the epigenetic codes might provide some, clue about heritable features that we observe in those worms.
Because you don't expect the DNA to be affected during those generations.
Because it's the same individual. It's
right.
like myself.
And you won't change it by spontaneous mutation. The
frequency is so low that in a population, you have all they are all like twins, let's say.
So can you give us like a very simple example of a trait that you followed across the generations?
Well, there are like in the beginning of my lab, we focused essentially on features that are related to germline and gametes, the cell that provide the sperm and the oocyte, just because this is the material that really give to the next generation some molecules. And so we study this weird phenomenon of worms that gradually become sterile after so many generations. And we discovered that in fact nothing is changing in terms of DNA mutation, but they inherit other type of molecules, epigenetic molecules that explain these features. But more recently, other labs have shown that even complex traits like behavior can be incredibly passed to the next generation. So this is very interesting. So the worms usually they eat bacteria, and usually they eat a healthy bacteria that we gave to them. But then if you have a plate and you let them choosing between two different foods, one that is pathogenic and is bad, and another one that is good, instinctively has an attraction for the bad food, like myself.
Yeah, exactly.
To sort of,
very similar to humans.
Yes. So then what happens is that, after he starts to experience his unhealthy food, he's very pathogenic and is sick. And basically, if you let them choose after certain hours that he experiences this food, when he chooses between the two food, he actually now learns to avoid the bad food and he goes to the good food. Okay? So this behavior, which is an acquired behavior after an experience that he never seen before in his life, right? And so you might ask, what happened to the babies of this worm that now experience those processes? And surprisingly, in fact, the babies of this worm, even though they never seen in their life before the pathogenic bad food or the good food, now they are not attracted anymore to the bad food, and they immediately go to the healthy food. Which means that, this behavior that has been, acquired during our life experience can also be somehow memorized somewhere and passed to the next generation. And this phenomenon lasts even for 4 or 5 generations, which is extremely long.
Fascinating.
So this is why now the key process is to really understand what are the molecules that can store this memory. And obviously this process is happening in the neuron, in the brain, or in the gut where they experience this food. So how these molecules that can memorize this information in the gut or in the brain are then transmitted to the sperm or the oocyte to be passed to the next generation. So these are the questions that we are right now studying, and we have very interesting observation that might lead to the explanation of how this phenomenon can be achieved.
Fascinating. I wonder if this can also be related to stress inherited.
Absolutely. So, in fact, other type of features, let's say, that can be heritable in worms, but also in other species. Now we have evidences that in mice something is happening the same. Is like, in fact, stress experiences that can be changes in the temperature or...
what about mother deprivation? Has anybody looked
starvation?
at that?
Absolutely.
And can it be
So
passed
this can have a direct impact on the next generation.
Wow.
And can have a what we call transgenerational impact if there are epigenetic molecules involved.
Wow.
Essentially, this is why it's fundamental to study simple organism where we can control environment pretty well,
sure.
and the genetics pool and the
we won't be able to do this in a longitudinal studies with humans, of course.
Once we apply those information in those organisms, then we can comparatively go into those higher organisms, maybe test specifically those responses.
Yeah, also to understand the molecular nature of this change.
But there are now much more evidences in a higher organism that the same process might be regulated similarly.
Wow, that's really fascinating. So, to change topics slightly,
or to share other things that we have in common.
Exactly. I also wanted to mention that me and Germano got together this time around at Red Hall because we were asked to participate to a workshop on drawings in science, and it was a beautiful event, mainly organized by Marie Dorigny, myself, and Renaud Chambrier from the Polytechnique here in Paris. Germano kindly accepted the invitation to speak, to give like a short presentation at the event. And the reason is because, among other things, Germano is also very much interested in the topic. He's an artist himself. And I'm gonna let it tell you a little bit more about why he gave this presentation, because I think illustrations in science is a beautiful topic that we might need to explore a little more. As a scientist, perhaps we need more of these platforms where we get together with the artists. There's a lot to be learned from each other, and I think communication is really important. There are not many people that fall in the two worlds, and I think probably Germano is one of them, so I'll let you talk a little bit about...
in fact, that's the reason why I also... I'm a good friend of the Institute, because we really try to foster, some interaction between the artists that every year are coming from New York or every part of the world in the Institute. And so we want to try to build with the Institut Pasteur some connection where the artists can experience what we do in the lab, and maybe, come up with something, some interactive
get inspired
works.
And,
yes. And this is in fact something that I have been doing in these past years, and that's why I participated to the workshop, because among some artists that we hosted in the lab, I met these two amazing people. They are one illustrator, Puya Abbasian, and Victor Coutard is a writer. And so basically, we met in a bar, drinking wine in Paris. And as usual, that's a good source of inspiration. And so talking about my work, they said, maybe why don't we do a book for kids on epigenetics? And I say, wow, I never thought about it. It sounds interesting, but we need to talk more. And so we spent a few occasions in the lab. I showed the works, and basically, it came out.
So now you can show what C. elegans is, right?
There is a drawing of C. elegans, which is very cute here.
Super cute.
And
I love the blue and pink, as the hermaphrodite.
So that's a really great example of. And it's a book for kids, but actually I enjoy so much reading the story. The story is very interesting. It's actually the story of two twins, Leon and Noel, who actually they are homozygous, so they are identical, but they don't feel they are identical. They they are two musicians. One likes jazz and one likes rock. They play in the same band, but they still ask themselves, why we are so different? And so they do a concert in Paris, which I think is here. There is a concert in Paris where the Professor Bonhomme,
That's you.
that's me, is a big fan of the band. In fact, I'm a fan of rock, and so he goes to the concert, and then he said, but actually I have an idea why, despite you are genetically identical, you might be different. And so he invited them to the lab. He showed the worms that are all identical, but they are not showing the same things. And so basically they learn something new, that maybe there is something else about genetics.
Fantastic.
That is also
I hope you're going to
epigenetics.
take up on a new project, because you seem very good at this.
Yeah,
No,
no, this
yeah,
was
but-
there was a
So
very good example.
I would just like to end this beautiful conversation with you, if you allow me,
yes.
to talk briefly about another passion we have in common, which is music.
Oh,
And so when I met Germano, I had the pleasure to share a lot of evenings together listening to music and everything. And of course, I didn't know that he was a professional drummer. So tell us a little bit about that.
that's
And the reason why we have this in common, rhythm is really in common, because on a side, I also have a passion for flamenco dance,
You are a
which
dancer?
I've been doing it for many, many years. So please tell us a little bit about what you're doing. Are you still playing?
Well, for my fortune, in fact, when I arrived to Paris, I discovered that basically I was not alone. I was not the only scientist playing music, and in fact, as you also mentioned, we all have a lot of interest, which is good. Basically, I discovered that many other people in my department were also playing music. So we decided to set up a band in Institut Pasteur. And now
I wish I could say the same about my.
we have a concert tomorrow. You are kindly invited. And so, essentially, it's a big fun, because this project is allowing people to from any different kind of PhD student, postdoc, senior scientist.
So it's a very inclusive.
Very inclusive.
Moment. Are you guys jamming on one type of music?
Yeah,
or-
I mean, I like rock and jazz and blues, but it depends on the people, and many people rotate. Some PhD student might leave, and then you.
What's
end up
your
with other people.
repertoire? Is like standard jazz songs, or you
right
going-
now it's more blues oriented.
Cool, I love that.
And so we have a rehearsal room in Pasteur, which is also something that we achieved to have it. I think it's a great project, because we spend a lot of time in the lab, but we should feed our passion too. So having a rehearsal room in the institute is something extremely important, because it's not easy in the city of Paris to find rooms to play, and so this is very comfortable after work. You jump into the music room and you can play. And you're welcome to come to dance, and maybe we can have a jam on
Ole, ole,
flamenco.
ole! So, fantastic. No, I would love to come. Let's see how the day unfolds tomorrow. But I think we can wrap it up and this wonderful conversation. Thank you so much for joining me here. I hope that there will be more occasions to be all together in this wonderful place. And thank you so much to Ray the Hall and to Marie, to James and
Thank you.
Arthur for assisting us with this incredible event. Thank you.